our technology has begun to eliminate the microbial dark matter. Just to start off, Chris, I was thinking about this very powerful robotic system that you have. One thing I was wondering was whether there are any constraints on microscopic observations in situ, or have you guys thought about it? And are there any reasons why direct high power, at least sort of 1000x microscopy is online?
Well, I think you hit it exactly. The context in which a lot of those analyses are performed is critical for interpretation. I used to always have a slide in my presentations that had bog mat and the Shroud of Turin, And, they both have human DNA, and they both have all these interesting microbes associated with them, but which artifact you're dealing with gives you an entirely different perspective on how to interpret that information. And so, so to your point, absolutely. I think it's just a matter of time and energy, and you could do that.
And one of the questions, actually, while we're there, was so you had mentioned that experiments can be done in situ. Have you considered, or have you done any in situ perturbation experiments where you would follow the same parcel of water over a period of time at a certain in a certain parcel of water?
Not any like as if we're doping the water in a particular way, but following natural processes. Eddies in the open ocean are a great example, and Garavandaneg is involved in some of this. So we're not really trying to perturb the system purposefully, but rather observe perturbations and the response of the microbial communities in kind. And it's a challenge because it's a space.
Big questions from the audience. Huh?
So this is just, my name is Johnathan, I'm here at ISB. Just to follow up on that particular question, this is for Nitin and anybody else who can answer this. So in your work, you do a lot of perturbation of the system, analysis, perturbation. And I'm wondering to what extent that perturbation cycle needs to occur, and to what extent one might be able to use the environmental or natural perturbations to infer regulatory networks and then infer behavior or predict behavior.
In principle, I think it's directly applicable. So the kind of data that Chris showed, if my understanding is correct, it's a group of microbes trapped in the vessel that are drifting, and you're measuring your transcriptomes?
Yeah, they're on the outside, and you're just drifting with them.
Yeah. So the key to this is knowing the source of the data that you're getting. A lot of metatranscriptomic data tends to be difficult to model because it comes from such a complex metagenome. And not everything is physically linked to the same genome necessarily. So some of the mechanistic principles that we use to model the system has to be reassessed because you can have genes compartmentalized in different membrane sacs. So there might be signaling, and you might find interesting associations, but this would be the most logical first step, is to take organisms that you know and then look to see how they perturb in spatiotemporal sense to natural environmental fluctuations. So I think it's doable.
So when you start to think about the interaction between the microbes, can you get, and I guess this is sort of even getting to Paul and then other people, can you get some idea from the CRISPRs and the phage sequences of what are the relationships of who has seen whom and how they're maybe building these interaction networks and themselves responding to it?
I mean, and there are other people here who probably have looked at it more, I don't know, but certainly if you get really deep data from a specific community and probably temporal data as well, you could probably start to use, I mean, it's not obviously just CRISPR information, but information about for people who don't know, CRISPRs are these parts of the genome of bacteria and archaea that are, in essence, an adaptive immune system to phage, and you can use them to recognize which phage individual strains might have encountered relatively recently. So you can compare different contigs to each other and look at the different CRISPR content. People have also done that with IS elements. So, I mean, I think you can use creative genomic analyses of many types to start to tease apart historical interactions among organisms, including just recombination, right, in the population genomic data, as well as the CRISPRs and IS elements, plasmids, et cetera.
I can add to that. So we looked at the differential regulation of CRISPR-associated genes, so Cases, and also the CRISPR elements themselves in Pyrococcus and Sulfolobus and so on. And even in absence of infection, you can see meaningful differential regulation in a way which reflects the spatiotemporal dynamics of infection, perhaps. And the schema of coupled environmental change is something that might go to all the way to infection as well, where the viruses are infecting at particular points in this during the physiological cycle of a cell, and maybe the organisms themselves are anticipating the CRISPR infection. So there's, one is knowing who's seen who, and the second is when they see each other. Would be a very interesting thing to look at.
So my first question is for Nitin. So you talked about co-regulation of genes. Have you thought about looking at co-regulation of transcripts in different organisms and try to see how they interact? And then also, what do you think of anti-correlation? You talk about co-regulation of the anti, and so that's.
We haven't looked at co-expression of genes, co-regulation of genes across cells, microbial organisms, but we have looked at tissues in humans where you have multiple cell types, and we begin to see signatures that dominate. And what we haven't looked at yet, but it's going towards what you're pointing out, is the expression of the genes themselves might be in different cell types, but you do see some coherent patterns that associate them, including microRNAs in different cell types and so on. Your second question was the anti-correlation. So that is captured in the regression step. So when you look at co-expression or to biclustering, you will find patterns that are correlated. But the question is, if you do have, say, a bifunctional regulator that is activating some genes and repressing some other genes, would you miss that? The answer is no, because when you do regression, you will find activator influences on one cluster and repressor on the other clusters.
So my second question is for Paul. So just out of curiosity, when you take a single microbe and sequence it, do you ever see extracellular DNA? Or do you actually treat it, right, with a nuclease before you do? Because I heard from some work that it's actually a lot of organisms, it release a lot of DNA. And so I'm just out of curiosity, do you ever see extracellular?
So our procedure is specifically designed to exclude extracellular DNA to the greatest extent we can. Now, that being said, I mean, if there's something attached to the cell and it survives our washing procedure, we'll sequence it, but we won't have a way to distinguish that from what was inside the cell. Now, if it's something that's copied from a gene in the cell, then I think that the question is more interesting at a functional level than a genomic level.
I have a question for Johnathan. How many genomes do you think you might have to sequence to figure out what a bacterial species is? Or will you figure it out at all? You have all these pan-genomes hanging around, they might merge or not merge. And then related to this, how long will it take that we find out what a gene is? I mean, one SNP, two SNPs, different gene, not?
Yeah, I mean, so the species concept in bacteria and archaea is a little more fluid, shall we say, than in plants and animals. But I think it's, from the studies that have been done so far on population genetics within a particular clade, it looks like you can treat the sort of core part of a bacterial and archaeal genome as roughly evolving like species do in other organisms. The problem is that the core is only, 20 to 40% of the genome, and there's lots of other parts of the genome that bounce around, like electrons or something to that effect. And we've been focusing on the deep branches in our project, so going from the bottom of the tree out to fill in. So we're not even remotely close to dealing with species. I mean, we're at at best the order level so far. But other groups have tried to say, let's do the same type of approach and fill in what is currently classified as, say, Salmonella or Escherichia or something to that effect. And my impression is that the pan genome, which is all the genes found within in essence, something named as the same species, is going to keep going on for a long time, because each individual strain in different parts of the world can pick up DNA relatively easily. But if you compare all of them to each other and reconstruct the history of an entire group, most of those genes that come in don't stick around over evolutionary time for a very long period. So studies by Vincent Daubin and a few other people have sort of suggested that a lot of the lateral gene transfer is recent, short-lived acquisition. So I think eventually we will have won't be the same as the species concept for plants and animals, but it's going to be a recombining population of closely related things that share DNA more frequently with each other than they do with other things. And the difference is that they can also acquire DNA from way the hell over there in the tree. So we have to sort of treat it more gently.
So a question for Chris. I presume that there are communities in the ocean that are stratified according to depth. And my question is there good evidence for that? And how are the stratifications continuous, or are they discrete, or
Absolutely, there are, based on a lot of ribosomal sequencing, some of the earliest studies would show that most of the microbes, which of course were not cultivated, are found in different strata. But it's a spectrum, and it's in constant motion. And so what is at 40 meters, for example, now may be at 20 meters an hour from now. And so it is phenomenally complex. And even in the surface, irrespective of depth, if you just open ocean, the oligotrophic North Pacific, people think of it as being pretty homogeneous. And Gary was part of this cruise just recently where, following some drifting instruments, there are changes in the microbial community on the scale of kilometers and an hour or a day. So I think it kind of blows us away at the diversity, not only in function and genomic capacity, but just in time and space. So absolutely, it's there.
I had a question for Paul, I guess. So I really like the SFB section of your talk, and I was curious whether or not you've only sort of looked at microbes from feces, or if you've tried to sort of use these single-cell approaches to get at evolution throughout the length of the gastrointestinal tract, or maybe over time.
Yeah, we haven't sampled at that detailed of a level yet. I just dropped my laser pointer. Don't worry.
I broke the chair.
And part of that's reflected in the fact that the single-cell sequencing technologies are pretty labor-intensive still. And so I think for the kinds of studies you're talking about, where we really do a large number of cells as a function of position in the GI tract, or do that kind of a study and then expand it on a time axis, we need to look to next generation single-cell sequencing platforms that can get to this thousands of cells level routinely. So that's something we can all look forward to.
Hi, I had a question for Dr. Baliga. I was wondering if what your thoughts are on gene clusters that are inherently not co-regulated, but if you could artificially, externally make them co-regulated, what your thoughts are on that. And on the converse, genes that are co-regulated, if you could make them decoupled by, for example, oxygen and temperature, and don't make them always go hand in hand.
Yeah. The coupling of genes or processes has been shown to be a fitness determinant as well. So if you artificially decouple, it results in loss of fitness. And that partly has to do with because the way energy resources and biochemical resources are all optimized for a certain process. And so when you decouple, I think you can't do it in a very localized way. And that's been a problem of synthetic biology. So if you try to decouple or bring in something new, you really put a tax on all the other other linked aspects of the system. So you really need to do systems level optimization where you begin to appreciate the cofactor needs of the new enzymes or the cofactor needs of the newly coupled systems that you have, and then change their regulation. And that gets to systems reengineering, which I think is a whole new field that doesn't exist right now, which will, I think, come about from the merger of synthetic and systems biology.
I have a question for Dr. Baliga as well. I come from a more ecological background myself, and I'm curious to hear your opinion and thoughts on the application of your techniques for identifying co-rems in ecological systems, where you might not have the ethical or practical abilities to perturb the systems, and also where levels of natural selection may not have given us such a nice machine.
I think it's doable if you have a spatio-temporally structured system where you can resample reproducibly, and you know the gradients you have in the environment, and you can track the, I mean, I think it, and I'm just speculating here. I've done a little bit of this, and other people have done, Eric Arm would have some good things to share in his talk as well. So you can look at spatio-temporal variations at the genome level to begin to understand how genetic variation tracks with environmental change. And you can couple that with transcriptomic analysis once you have the foundational information, and you could apply similar strategies. The challenge in environmental analysis at the level that I spoke comes from the complexity of the microbial communities, and knowing what signal is from noise is at the heart of it. So I think much of the initial work will happen in environments which are much reduced naturally because of extreme conditions. Species diversity is low, the spatio-temporal structure. So I think it's doable. Theoretically, I don't see a reason why it can't be done. And I think the co-rems that you're referring to in the ecological sense would, I think, potentially include, I think it was a question earlier, genes from multiple organisms, because you'll have long range, depending on the scale we're talking of, you'll have long range associations and functions across cells.
So a question for Dr. Eisen. You keep talking about a tree, but coming from a computer science background, it seems like you're dealing with a DAG, with horizontal gene transfer. At the very smallest scales, you can have cross-links and so forth. So yes, well, on a large scale, it looks like a tree. When you're getting down to that small scale, it's a different structure. What sort of, how does that affect your thinking about the way to do this if you've basically got a mathematical model that seems to be very centered on a tree as opposed to a DAG?
Yeah, so what I tried to say at the beginning was when I use phylogeny, I use it to refer to the history, not a tree per se. And so for the studies of plants and animals for years, people have been using bifurcating tree representation, even though we know that within populations, within species, it's completely reticulated in all the sexually reproducing organisms, and that leads to all sorts of challenges in interpretation, like ancestral polymorphisms, where a single allele will be shared between individual humans and chimps and not between multiple humans within the population. There are hybridization events. There are all sorts of reticulations. There's even lateral gene transfers in plants and animals. And so nevertheless, depending on what depth you're looking at for your particular question, bifurcating trees can sometimes be still a useful model in plants and animals. And so the challenge with bacteria and archaea is really the extent to which there is lateral gene transfer at different depths and what the model is that you should use to represent history. That's what I mean by phylogeny. So actually in many of the things we're doing now that we're doing with Aaron Darling, who's in my lab, and that multiple people are trying to do is actually having a Bayesian or likelihood model of reticulation as part of your analysis. And so you include the network as opposed to a tree. But it's still important to include the history behind biological objects. It's just you do have to have it helps to have a more robust model of what that history actually is. And it depends on the question how much it seems to matter. So for certain types of questions, it seems to not matter that much. And maybe getting back to Per Bork's question, which I didn't answer the second part, if you're looking at gene evolution as opposed to species evolution, and you want to do certain things with gene evolution, like divide genes into superfamilies, phylogeny is a pretty good way to do it. If you want to look at, identifying sort of haplotypes, phylogeny is not a good way to do it. You have to use recombinational reticulation models. So I just sort of glossed over that because I wanted to represent history as the key thing, but not specifically bifurcating trees. So it's a very good point.
Take one more question.
Sure. David Baumler, University of Wisconsin-Madison. This is a two-part question for Professor Baliga and also Dr. Eisen. So ancient halophilic microbes have been isolated from salt inclusions dating back over 100 million years. Have you thought about maybe starting to study some of these ancient microbes by building computational models of them?
I used that as an opener for my talks. I never thought of using it, but there was a lot of controversy surrounding it. Maybe
I don't believe the findings.
They shut me off. Oh, sorry. Professor Eisen, so you started to talk about these very core proteins being conserved across the board in a lot of your phylogenetic tree of life studies. Have you thought about using these core sets to start to build maybe ancestral core or ancestral state models for entire families, or even going back to phylums where we could start to go dating back millions, possibly billions of years?
Yeah, I mean, we've done a little of this. Actually, Eric Alm, who's going to be talking, might even talk. He's done much more on this type of thing. So we haven't focused that much on trying to infer the history of protein families within particular lineages. Other people have used genomic data, including our data, to do those models and try and I don't want to scoop Eric's talk, but compare, for example, the protein family history with geological history and look at the evolution of features in those families with the history of the Earth. So we have not done that much of that, but a lot of other people have done really interesting stuff on that.
Thank you.
Thank you. Well, thank you very much for coming to the session, and thanks to the speakers.